Hey everyone, welcome to this deep dive, especially for Mr. Martin's IB biology class. Have you ever just stopped and thought, why do living things even bother reproducing? I mean, it seems so basic, but it really drives everything in biology.
That's actually a really great question to kick things off. It gets right to the heart of it, doesn't it? Keeping life going, continuity, but also allowing for change, for evolution. Reproduction is how life manages both.
Exactly. So for this deep dive, we're really zeroing in on reproduction, specifically tailored for you, Mr. Martin's class. The idea is to get the essential stuff, the core knowledge you need from your IB syllabus without feeling totally overwhelmed. We're pulling from your textbooks, notes, all that good stuff.
Yeah, our mission today is basically to unpack the main principles. We'll look at the fundamentals like asexual versus sexual, but also dig into the specifics for humans and flowering plants, which are, key examples in your course. We'll highlight some key differences and maybe some surprising facts along the way.
All right, sounds like a plan. Let's jump in. Asexual versus sexual reproduction. What's the fundamental difference we need to grasp first?
Well, simply put, asexual reproduction is just one parent producing offspring, and these offspring are genetically identical to the parent. They're essentially clones.
Okay, clones. So how does that work? How does one organism just split into two identical ones without any, mixing of genes?
It's fundamentally about cell division. The parent cell duplicates its DNA and all the important internal bits, the organelles, and then it divides. The key point is there are no specialized sex cells, no gametes like sperm and eggs fusing together. It's much more direct.
That sounds really efficient. Like if you're perfectly happy where you are in a stable environment, just making copies seems like a good strategy.
Precisely. If the parent's traits are well suited to the current conditions, making exact copies passes on those successful traits directly. Plus, it's usually faster and it saves the energy you'd otherwise spend finding a mate. Populations can boom quite quickly.
Right. But there's always a catch, isn't there? What's the downside to everyone being the same?
The big one is the lack of genetic variation. If the environment changes, maybe a new disease pops up, or the temperature shifts, a population of clones is incredibly vulnerable. They all share the same weaknesses. There's no diversity for natural selection to act upon, no individuals who might happen to have a trait that helps them survive the new challenge.
Okay. So no real flexibility if things get tough.
Yeah.
Got it. Can you give us some examples? Where do we see asexual reproduction happening?
Oh, it's everywhere, really. Mitosis, the process your own body cells use to divide for growth and repair Mr. Martin's class. Think about how a cut on your skin heals. That's a form of asexual reproduction at the cellular level. Then you have binary fission in prokaryotes like bacteria. It's similar, but simpler because bacterial cells aren't as complex.
Simpler cells, simpler splitting. Makes sense.
Yeah.
What else?
Well, there's budding. You see this in yeast, which are fungi, and also in some simple animals like Hydra. It's like a small outgrowth, a bud, forms on the parent, and eventually detaches to become a new individual. It's not always an equal split like fission.
Unequal splitting. Interesting. Keep going.
Then fragmentation. Some organisms, like certain sea stars and worms, can regenerate a whole new organism from a broken off piece. Imagine a starfish losing an arm and that arm growing into a completely new starfish.
Wow, okay, that's pretty cool. An arm becoming a whole new individual.
It really is. And then there's parthenogenesis. That's when an egg develops into an embryo without being fertilized by sperm. It happens in quite a few insects, like bees, ants, wasps, and even some vertebrates, like certain lizards and fish.
No fertilization needed. Nature has all sorts of tricks.
It certainly does. And let's not forget plants. Vegetative propagation is basically asexual reproduction in plants. Think about taking a cutting from a plant, sticking it in soil, and it grows into a whole new plant. That's using stems, roots, or leaves from the parent.
So many ways to make copies. Okay, let's switch gears. What about the other side of the coin? Sexual reproduction.
Right, sexual reproduction. This involves two parents, each contributing genetic information in the form of specialized cells called gametes, typically sperm from the male, and an egg, or ovum, from the female. These gametes fuse together during fertilization.
And the result is offspring that are genetically unique, right? Not clones.
Exactly. The key is that the gametes are haploid.
Haploid, meaning they have half the normal number of chromosomes.
Correct. Each parent produces these haploid gametes through a special type of cell division called meiosis. Meiosis halves the chromosome number. Then, when the sperm and egg fuse during fertilization, the normal diploid chromosome number is restored in the offspring, the zygote. But crucially, the offspring's genetic makeup is a new combination inherited from both parents.
Okay, so meiosis creates the potential for mixing, and fertilization does the mixing. What's the big advantage of going through all this trouble?
The number one advantage is genetic variation. By shuffling genes from two different parents, you get offspring with unique combinations of traits. This diversity is incredibly important for adaptation. If the environment changes, there's a much higher chance that some individuals in the population will have traits that allow them to survive and reproduce successfully. It's like hedging your bets against future uncertainty.
So variation is the key benefit,
Yeah.
but it definitely sounds like more effort than just splitting.
It absolutely is. The downsides include, well, needing two parents, which isn't always easy. You need to produce gametes, get them together for fertilization, often involving complex mating behaviors or mechanisms. It generally costs more energy and time, and population growth is usually slower compared to asexual methods.
Okay, so quick summary. Asexual is great for stability and speed. Sexual is better for variation and adapting to change. That makes sense. Let's zoom in on humans now, starting with the male reproductive system. What's its main role?
The male system is designed primarily to produce sperm, then maintain and transport it within a fluid called semen. It also needs a way to deliver that semen into the female reproductive tract for internal fertilization, and it's responsible for producing the primary male sex hormone, testosterone.
Okay, let's trace the path. Where does it all begin? What are the key structures?
It starts in the testes, or testicles. These are the primary reproductive organs where sperm are produced in a process called spermatogenesis. They also produce testosterone. The testes are located within the scrotum.
And the scrotum's important
Because it hangs outside the main body cavity, keeping the testes slightly cooler than core body temperature. This lower temperature, maybe 2-3 degrees Celsius cooler, is actually optimal for sperm development. Too warm, and sperm production can be impaired.
Right, temperature control. Okay, so sperm are made in the testes. Where do they go next?
After being produced, they move into the epididymis. This is a long, coiled tube located on the back of each testes. Here, the sperm mature, they gain motility, the ability to swim, and they're stored until ejaculation.
Maturation and storage. Then, during ejaculation.
During ejaculation, sperm are propelled from the epididymis through a tube called the sperm duct, or vas deferens. This duct travels up from the scrotum, loops around the bladder, and eventually joins the urethra.
But it's not just sperm traveling, right? It gets mixed with fluids.
Exactly. As the sperm travel through the ducts, several glands add fluids to create semen. The seminal vesicles, located behind the bladder, contribute the largest volume of fluid. It's thick, alkaline, meaning it helps neutralize the acidic environment of the female vagina, and rich in fructose, which provides energy for the sperm. It also contains mucus and proteins.
Okay, seminal vesicles add the bulk and fuel. What about the prostate gland?
The prostate gland sits just below the bladder and surrounds the urethra where the sperm ducts join it. It secretes another alkaline fluid, also containing nutrients, into the urethra. The prostate's muscular contractions also help propel the semen forcefully out during ejaculation.
So sperm plus fluids from seminal vesicles and prostate equals semen. Any other contributors?
There are also the bulbourethral glands, or Cowper's glands, tiny glands below the prostate. They secrete a clear mucus just before ejaculation, which helps lubricate the urethra and neutralize any acidic urine residue.
Got it. And the final pathway out?
Is the urethra. This tube runs from the bladder through the penis to the outside. Importantly, it serves a dual purpose in males, carrying both urine during urination and semen during ejaculation, though not usually at the same time.
And the penis itself?
The penis is the external organ that facilitates internal fertilization. It contains erectile tissue that fills with blood during arousal, causing an erection, which allows semen to be deposited into the female vagina. And we already mentioned the scrotum holding the testes.
Right. Now, you mentioned sperm need to be motile. How does their structure compare to the egg cell? They sound very different.
Oh, vastly different, designed for completely different roles. Sperm are incredibly small, highly specialized for motility and delivering genetic material. They consist of a head containing the haploid nucleus and capped by the acrosome.
Acrosome, that sounds important.
Yes, the acrosome contains enzymes crucial for penetrating the egg's outer layers. Then there's a midpiece packed with mitochondria to generate ATP, the energy currency, to power the flagellum.
The flagellum being the tail.
Exactly, the long tail that whips back and forth to propel the sperm. They have very little cytoplasm and virtually no stored food reserves, just enough fuel for the journey. And they are produced in enormous numbers.
Millions, right. compared to the egg?
Compared to the egg or ovum, which is one of the largest cells in the human body, the ovum is non-motile. It can't move on its own. Its main job, besides carrying the female haplonucleus, is to provide nutrients for the very early stages of embryonic development if fertilization occurs. So it has a large volume of cytoplasm rich in yolk-stored food.
Big, nutrient-rich, and stationary versus tiny, streamlined, and mobile. And typically only one egg is released at a time.
Usually, yes, just one mature ovum is released per menstrual cycle. It's surrounded by protective layers like the jelly-like zona pellucida and an outer layer of follicle cells called the corona radiata.
Okay, that sets the stage nicely. Let's move to the female reproductive system. What are its primary functions?
The female system is multifaceted. It needs to produce ova, mature them, and release them for potential fertilization. It also has to provide the environment to receive sperm, support fertilization, and then protect and nourish the developing embryo and fetus throughout pregnancy until birth. And, like the male system, it produces key sex hormones, primarily estrogen and progesterone.
Okay, let's walk through the main structures. Where are the eggs produced?
The eggs, or ova, develop within the ovaries. Females are born with all the potential eggs they'll ever have stored as immature follicles. Each month after puberty, typically one follicle matures and releases an ovum. This process is ootogenesis. The ovaries are also the main producers of estrogen and progesterone.
And once an egg is released from the ovary, where does it go?
It's usually swept into the adjacent oviduct, also known as the fallopian tube. There's one associated with each ovary. The oviduct is lined with cilia, tiny hair-like structures that help move the ovum along towards the uterus. Crucially, the oviduct is usually where fertilization happens.
Okay, so fertilization site is the oviduct. Then, assuming fertilization occurs, the journey continues to the home.
The uterus, often called the womb, this is a strong, muscular organ with a thick inner lining called the endometrium. The uterus is where the fertilized egg, now developing into an embryo, will implant and grow for the entire duration of pregnancy. The endometrium is rich in blood vessels and glands to nourish the developing fetus.
And the endometrium changes throughout the month.
Yes, it undergoes cyclical changes, thickening and preparing for implantation under the influence of hormones. If pregnancy doesn't occur, most of the endometrium is shed during menstruation.
We'll come back to that cycle. What connects the uterus to the outside?
At the lower, narrow end of the uterus is the cervix, which acts like a gateway. It opens into the vagina.
And the vagina's roles.
The vagina is a muscular tube connecting the cervix to the outside of the body. It serves several functions. It receives sperm during sexual intercourse. It's the passageway for menstrual flow out of the body. And it serves as the birth canal during childbirth. The external female genital structures are collectively known as the vulva.
Okay. Now let's tackle that cycle you mentioned, the menstrual cycle. It sounds complex, involving both the ovaries and the uterus.
It is quite an intricate interplay, yes. You have two cycles happening concurrently, synchronized by hormones. The ovarian cycle, which involves the maturation and release of the egg and the formation of the corpus luteum, and the uterine cycle, which involves the changes in the endometrium.
Hormones again, the master controllers. Which ones are the key players here?
The main players involve the brain and the ovaries. The hypothalamus in the brain releases GnRH, which signals the anterior pituitary gland, also in the brain, to release two crucial hormones: follicle-stimulating hormone, FSH, and luteinizing hormone, LH.
FSH and LH, what do they do specifically?
FSH, as the name suggests, stimulates the growth and development of ovarian follicles at the beginning of the cycle. As these follicles grow, they start producing estrogen. LH levels surge dramatically around the middle of the cycle, and this LH surge is the primary trigger for ovulation, the release of the mature egg from the dominant follicle. LH also promotes the development of the corpus luteum from the ruptured follicle after ovulation.
And the ovarian hormones, estrogen and progesterone,
Yeah.
how do they fit in?
They're essential. Estrogen, produced mainly by the developing follicles, has several roles. It promotes the growth and thickening of the endometrium, preparing it for potential implantation. It also feeds back to the pituitary, initially inhibiting FSH, but then at high levels actually triggering that LH surge. Progesterone is produced mainly by the corpus luteum after ovulation. Its main job is to maintain the thickened endometrium, making it receptive and supportive for an embryo. It also strongly inhibits the release of GnRH, FSH, and LH, preventing new follicles from developing during the second half of the cycle.
Wow, it's a real dance of hormones with positive and negative feedback loops. Can you walk us through the phases, say, starting from day 1 of menstruation?
Okay. Day 1 is the start of menstruation, the shedding of the uterine lining. This corresponds to the beginning of the follicular phase in the ovary. FSH levels start to rise slightly, stimulating several follicles to grow. As they grow, they produce increasing amounts of estrogen.
And this rising estrogen affects the uterus.
Yes, the rising estrogen causes the endometrium, which was just shed, to start rebuilding and thickening again. This is the proliferative phase of the uterine cycle.
Okay, follicles grow, estrogen rises, endometrium rebuilds. What happens next?
Usually, one follicle becomes dominant and continues to mature, all the others regress. This dominant follicle produces very high levels of estrogen. When estrogen reaches a sustained peak, it exerts positive feedback on the pituitary, causing that massive surge in LH and a smaller surge in FSH.
An LH surge.
Yeah.
And that means
Ovulation. Roughly 24-36 hours after the LH surge begins, the mature follicle ruptures, releasing the secondary oocyte, the egg. This typically happens around day 14 of a 28-day cycle.
egg released. What happens to the follicle left behind?
The remnants of the ruptured follicle transform into a structure called the corpus luteum, thanks to the influence of LH. This marks the beginning of the luteal phase of the ovarian cycle.
And the corpus luteum starts making hormones.
Exactly. It becomes a temporary endocrine gland, secreting large amounts of progesterone and also some estrogen. This progesterone is crucial. It acts on the endometrium, causing it to become even thicker, more vascularized, and glandular entering the secretory phase. It's essentially making the uterus perfectly receptive for an embryo to implant.
So the uterus is ready and waiting. What happens if fertilization doesn't occur?
If there's no fertilization and implantation within about 10-14 days after ovulation, the corpus luteum starts to degenerate. It basically runs out of steam without hormonal support, which would come from an implanted embryo.
And when the corpus luteum degenerates,
Progesterone and estrogen levels plummet. Without the hormonal support, especially from progesterone, the thickened endometrium breaks down. Blood vessels constrict, then rupture, and the tissue is shed. This shedding is menstruation, marking the start of the next cycle.
and the drop in hormones also signals the brain to start over.
Precisely. The low levels of estrogen and progesterone remove the inhibition on the hypothalamus and pituitary, allowing FSH levels to start rising again, stimulating a new cohort of follicles, and the whole cycle begins anew.
Okay, that provides a clear picture of the cycle when pregnancy doesn't happen. Now, what if it does? What if a sperm successfully fertilizes that released egg? Where does this usually happen?
As we mentioned, fertilization typically occurs in the outer part of the oviduct, or fallopian tube. Sperm have to travel all the way up from the vagina, through the uterus, and into the oviducts to meet the egg.
That's quite a journey for the sperm. What happens when one finally reaches the egg?
Well, first it has to get through the outer layers. Remember the corona radiata cells and the zona pellucida. The acrosome reaction helps here. Enzymes released from the sperm's acrosome digest a path through these layers.
Okay, enzymes clear the way. Then what?
When a sperm successfully penetrates the zona pellucida and reaches the egg cell membrane, their membranes fuse. The sperm nucleus containing the father's genetic material enters the egg's cytoplasm. The sperm's tail and mitochondria typically degenerate inside the egg. Interesting note. This is why we inherit mitochondrial DNA almost exclusively from our mothers.
Ah, maternal inheritance of mitochondria. Cool. So sperm nucleus is inside. What next?
The entry of the sperm triggers the completion of meiosis II in the egg, forming the mature ovum nucleus. Then, the nuclear membranes of both the sperm and the ovum break down, allowing their chromosomes to mingle. This forms the diploid zygote, the first cell of the new individual, ready for its first mitotic division.
Now, I remember learning it's critical that only one sperm fertilizes the egg. Why is that, and how does the egg ensure this?
Yes, preventing polyspermy fertilization by more than one sperm is absolutely essential. If it happened, the resulting zygote would have an abnormal number of chromosomes, triploid or more, and wouldn't be viable. The egg has mechanisms to block this. We mentioned the acrosome reaction allowing penetration, but there's also a rapid response upon fusion.
What's the first line of defense once a sperm fuses?
Almost immediately upon fusion, there's a change in the electrical potential across the egg membrane called the fast block to polyspermy, which makes it harder for other sperm to fuse. But this is temporary in mammals.
So there's a more permanent block.
Yes, the cortical reaction, which is the slow block. When the first sperm fuses, it triggers the release of calcium ions within the egg cytoplasm. This calcium wave causes tiny vesicles just under the egg membrane, called cortical granules, to release their contents by exocytosis into the space between the egg membrane and the zona pellucida.
And what do these cortical granules do?
Their enzymes modify the zona pellucida, causing it to harden and also altering the sperm receptor proteins within it. This makes the zona impenetrable to any other sperm that arrive later. It effectively seals the deal.
So, first one in to pull air, triggers the door to slam shut. Clever.
Okay.
Fertilization complete, polyspermy blocked, we have a zygote. What happens to it?
The zygote immediately begins a series of rapid mitotic cell divisions called cleavage. It divides into 2 cells, then 4, then 8, and so on, forming a solid ball of cells called a morula. All this is happening while it's still traveling down the oviduct towards the uterus.
So it's dividing but not really growing bigger overall.
Exactly. The cells become progressively smaller with each division during cleavage. As division continues, a fluid-filled cavity forms within the ball of cells, transforming the morula into a blastocyst. The blastocyst consists of an inner cell mass, which will develop into the embryo itself, and an outer layer called the trophoblast.
And the blastocyst is what reaches the uterus.
Yes. By about 6-7 days after fertilization, the blastocyst reaches the uterus and begins the process of implantation. The trophoblast cells play a key role here, interacting with the receptive endometrium and embedding the blastocyst into the uterine wall.
Implantation. And this is where the connection between mother and baby starts to form. The placenta.
Precisely. Following implantation, the placenta develops. It's a remarkable organ formed from both fetal tissues, derived from the trophoblast, and maternal tissues from the endometrium. It becomes the vital interface for exchange between mother and fetus throughout pregnancy.
What exactly gets exchanged across a placenta?
It's a two-way street. Nutrients, oxygen, antibodies, and water pass from the mother's blood into the fetal blood. Conversely, waste products like carbon dioxide and urea pass from the fetal blood into the mother's blood so her body can eliminate them.
How does it manage this exchange so efficiently?
The placenta develops an incredibly large surface area for exchange. This is achieved through numerous finger-like projections called chorionic villi, which contain fetal capillaries and extend into spaces filled with maternal blood. The barrier between the fetal capillaries and the maternal blood is very thin, typically just a few cell layers thick, allowing for rapid diffusion.
But the blood itself doesn't mix, right?
Correct. That's a crucial point. Maternal and fetal blood circulate in close proximity within the placenta, but are kept separate by the placental barrier. This is important, partly because the mother and fetus might have incompatible blood types. Mixing could cause an immune reaction.
So substances have to cross this barrier. How do they do that? Different methods for different molecules.
Yes, various transport mechanisms are used. Gases like oxygen and carbon dioxide move by simple diffusion down their concentration gradients. Glucose, the main energy source, moves by facilitated diffusion using protein transporters. Water moves by osmosis. Amino acids, vitamins, minerals, and antibodies, providing passive immunity to the fetus, often require active transport or endocytosis, which uses energy.
And the placenta has another job too, doesn't it? Related to hormones.
Absolutely. Early in pregnancy, the implanted embryo, specifically the trophoblast, starts producing human chorionic gonadotropin, or HCG. This is the hormone detected by pregnancy tests. HCG's main role is to maintain the corpus luteum in the ovary, preventing it from degenerating.
So HCG keeps the progesterone flowing from the corpus luteum.
Exactly, for the first few weeks. Progesterone is vital for maintaining the endometrium and preventing uterine contractions. However, the placenta gradually takes over hormone production. By the second trimester, the placenta itself is producing large amounts of progesterone and estrogen sufficient to maintain the pregnancy, and the corpus luteum is no longer needed and regresses.
So the placenta becomes the main hormone factory for the rest of the pregnancy.
Okay, that's a good overview of human reproduction. Shall we switch kingdoms now and talk about flowering plants?
Let's do it. Flowering plants, or angiosperms, are incredibly diverse, and their reproductive structures, the flowers, are fascinating adaptations for sexual reproduction.
Okay, so the flower is the main reproductive unit. Can you break down the typical parts of a flower and their roles? Like if we were annotating a diagram for Mr. Martin's class.
Sure. Starting from the outside, you often have sepals. These are typically green and leaf-like, and their main job is to protect the flower bud before it opens. Inside the sepals are the petals, which are often brightly colored or scented to attract pollinators like insects or birds.
Sepals for protection, petals for attraction. What about the reproductive parts themselves?
Okay, the male parts are collectively called the stamen. Each stamen usually consists of a stalk-like filament supporting an anther at the top. The anther is where pollen grains containing the male gametes are produced.
Stamen is male. Anther makes pollen. And the female part? The female part is the carpel, or sometimes called the pistil, which can be one or several fused carpels. It typically has three regions. The stigma at the top, which is often sticky or feathery to receive pollen, the style, a stalk connecting the stigma down to the ovary, and the ovary at the base, which contains one or more ovules. Inside each ovule is where the female gamete, the egg cell, develops.
Carpel is female. Stigma receives pollen. Style connects. Ovary holds ovules with eggs.
Got it.
Any other common parts?
Many flowers also have nectaries, glands that produce sugary nectar, which serves as a reward for pollinators. The arrangement and specific features of these parts vary hugely depending on the plant species and its pollination strategy.
Okay, so analogous to sperm and eggs, plants have pollen and eggs inside ovules. How are these gametes actually formed in plants? Let's start with pollen.
Pollen production, or microsporogenesis, happens within the anthers. Inside specialized chambers, diploid cells called microspore mother cells undergo meiosis. Each meiosis produces 4 haploid microspores.
Meiosis again, having the chromosomes. What happens to these microspores?
Each haploid microspore then undergoes mitosis, but usually without full cell division, to develop into a pollen grain. A mature pollen grain typically contains 2 nuclei within a single cytoplasm, enclosed by a tough outer wall. These are the tube nucleus and the generative nucleus.
2 nuclei in one pollen grain. What do?
The tube nucleus will control the growth of the pollen tube after pollination. The generative nucleus is the one that will divide again, usually later during pollen tube growth, by mitosis to produce 2 male sperm cells. So ultimately, one pollen grain delivers 2 sperm cells.
2 sperm cells per pollen grain. Interesting. Now, what about the egg cell formation in the ovule?
That process is called megasporogenesis and megagametogenesis. Inside the ovule, a diploid megaspore mother cell undergoes meiosis, producing 4 haploid cells. Usually 3 of these degenerate, and only 1 survives as the functional megaspore.
Okay, one surviving haploid megaspore, then what?
This megaspore then undergoes 3 rounds of mitosis, but again, without the cytoplasm fully dividing between each nucleus. This results in a large structure called the megagametophyte, or more commonly, the embryo sac. A typical mature embryo sac contains 8 haploid nuclei arranged in a specific way within 7 cells.
8 nuclei. What are the important ones?
The most important ones for fertilization are the egg cell itself, located near an opening called the micropyle, and 2 other nuclei called the polar nuclei, which are usually located together in the large central cell of the embryo sac.
Okay, so we have pollen grains with sperm potential and ovules with an egg and polar nuclei. How do they get together? Pollination, right?
Exactly. Pollination is simply the transfer of pollen from an anther to a stigma. It's the crucial first step. It can be self-pollination, where pollen lands on the stigma of the same flower or another flower on the same plant, or cross-pollination, where pollen travels from one plant to a different plant of the same species.
And plants have evolved amazing ways to achieve this, often involving partners.
Absolutely. Many rely on biotic pollinators, insects, birds, bats, etc. These flowers often have specific adaptations: bright colors, strong scents, nectar guides, specific shapes to fit the pollinator. The pollen itself might be sticky or spiny to adhere to the pollinator, and the stigma large and sticky to catch it.
Like bees visiting colorful flowers for nectar. What about abiotic pollination?
Wind pollination is also very common, especially in grasses and many trees. These flowers are typically less showy, often lacking petals and scent. They produce vast quantities of lightweight, smooth pollen that's easily carried by the wind, and they often have large, feathery stigmas to effectively trap airborne pollen. Water pollination occurs in some aquatic plants.
Plants have also developed ways to encourage cross-pollination over self-pollination, haven't they? Why is that preferred?
Cross-pollination increases genetic diversity, which, just like in animals, enhances adaptability. Self-pollination can lead to inbreeding depression over time, so plants have several mechanisms to promote crossing. One common way is having different timing for the maturation of male and female parts within a flower. Either the anthers mature and release pollen before the stigma is receptive, protandry, or the stigma becomes receptive before the pollen is shed, protogeny.
Temporal separation. What else?
Some plants have structural arrangements that make self-pollination difficult. Others are dioecious, meaning they have separate male plants, producing only staminate flowers, and female plants, producing only carpellate flowers, making self-pollination impossible. Monoecious plants have separate male and female flowers on the same individual.
And I think you mentioned self-incompatibility before.
Yes, that's a widespread genetic mechanism. The stigma style can recognize pollen from the same plant or genetically similar plants, and actively inhibit pollen germination or pollen tube growth, ensuring fertilization only occurs with pollen from a different genetic individual.
Okay, so pollen successfully lands on a compatible stigma. What's the next step?
Germination. The pollen grain absorbs moisture from the stigma and glows a pollen tube. Guided by chemical signals from the pistil, this tube grows down through the style towards the ovary and ultimately enters an ovule, usually through the micropyle opening.
And the sperm cells travel down this tube.
Yes, remember the generative nucleus inside the pollen grain. As the pollen tube grows, or sometimes just before, the generative nucleus divides by mitosis to form the two haploid sperm cells. These two sperm cells travel down the pollen tube to reach the embryo sac within the ovule.
Two sperm cells arriving at the embryo sac. This leads to something unique in flowering plants, right?
Yes, this is where the famous double fertilization occurs, a hallmark of angiosperms.
Double fertilization. What gets fertilized twice?
Okay, so the pollen tube releases the two sperm cells into the embryo sac. One sperm cell fuses with the egg cell. This is standard fertilization, forming the diploid 2N zygote, which will develop into the embryo.
Right, sperm plus egg equals zygote. What does the second sperm do?
The second sperm cell migrates to the central cell of the embryo sac and fuses with the two polar nuclei that are already there. Remember those?
Ah, yes, the two polar nuclei in the middle. So sperm N fuses with two polar nuclei, N plus N. What does that create?
It creates a triploid 3N nucleus. This triploid nucleus develops into the endosperm, which is a nutrient-rich tissue.
Triploid endosperm. And its job is?
Its primary role is to serve as the food supply for the developing embryo, either during seed development or later during germination. So, double fertilization results in both the embryo, the new plant, and its packed lunch, the endosperm.
That's incredibly efficient. One event fertilizes the egg and creates the food source. Okay, fertilization done. What happens to the ovule and the ovary after this?
After double fertilization, major transformations occur. The fertilized ovule develops into the seed. The seed contains the diploid embryo, the triploid endosperm, or remnants of it if it's absorbed by the embryo's seed leaves, the cotyledons, and a protective outer seed coat derived from the ovule walls.
Ovule becomes the seed.
Yeah.
And the ovary surrounding the ovule.
The ovary wall, and sometimes other floral parts, develops and matures into the fruit. The fruit's main function is typically to protect the seeds and aid in their dispersal.
Seed dispersal. Moving away from the parent. Why is that so important?
It reduces competition between the parent plant and its offspring, and among the offspring themselves, for resources like light, water, and nutrients. It also allows the species to colonize new habitats.
And plants use various agents for dispersal, similar to pollination.
Absolutely. Fruits and seeds have evolved countless adaptations for dispersal. Wind dispersal relies on lightweight structures, wings, or parachutes, like maple keys or dandelion seeds. Water dispersal uses buoyant fruits or seeds, like coconuts.
And animals are major players too.
Huge players. Many plants entice animals with fleshy, edible fruits. The animal eats the fruit, moves away, and later excretes the seeds, often in a nice little pile of fertilizer. Other seeds have hooks or barbs, like burrs, that attach to animal fur or feathers and get carried away. Some, like acorns, are buried by animals like squirrels and forgotten. And some plants use ballistic dispersal. The fruit dries and splits open explosively, flinging the seeds.
So many strategies. Okay, a seed lands in a suitable spot. What triggers it to actually start growing? What is germination?
Germination is the process where the embryo inside the seed resumes growth and emerges to form a seedling. It requires a specific set of environmental conditions.
Or the essential conditions?
Generally, three things are crucial. Water is needed to rehydrate the seed tissues, which activates metabolic enzymes and softens the seed coat. Oxygen is required for aerobic cellular respiration to provide the energy, ATP, needed for growth. And a suitable temperature range is necessary because germination relies on enzyme activity, which is temperature sensitive. Some seeds also have specific requirements like light or periods of cold stratification.
Assuming conditions are right, what happens inside the seed?
The embryo becomes metabolically active. It starts using the stored food reserves, either from the endosperm or from the cotyledons. The embryonic root, called the radicle, is usually the first part to emerge, anchoring the seedling and absorbing water. Then the embryonic shoot, the plumule, which includes the embryonic leaves and stem, grows upwards towards the light.
And a new plant begins its life. Fantastic. Okay, we've covered the core reproduction in humans and plants. For the HL students in Mr. Martin's class, let's briefly touch on puberty and menopause in humans. What drives puberty?
Puberty is that whole transition period from childhood to sexual maturity. It's orchestrated by hormones, starting with the hypothalamus in the brain, increasing its secretion of gonadotropin-releasing hormone, GnRH.
Right. GnRH kicks things off. What does it trigger?
GnRH acts on the anterior pituitary gland, causing it to ramp up the release of LH and FSH. These pituitary hormones then travel to the gonads, testes in males, ovaries in females, and stimulate them to significantly increase the production of sex hormones.
Testosterone in males, estrogen and progesterone in females.
Exactly. And it's these surging levels of sex hormones that drive the development of secondary sexual characteristics, like voice deepening in boys, breast development in girls, growth spurts, maturation of the reproductive organs, and the onset of fertility.
Okay. And then much later in life, for females, there's menopause. What's happening hormonally there?
Menopause marks the permanent end of menstruation and fertility, typically occurring somewhere between the mid-40s and mid-50s. The fundamental cause is the depletion of ovarian follicles. As a woman ages, the number and responsiveness of follicles in her ovaries decline drastically.
Fewer follicles mean less.
Less estrogen and progesterone production by the ovaries. As estrogen levels fall, they no longer exert the same negative feedback on the pituitary gland. Consequently, the pituitary releases more FSH and LH in an attempt to stimulate the ovaries, so FSH levels become characteristically high after menopause.
And the lack of estrogen causes the symptoms associated with menopause.
Yes, the decline in estrogen is responsible for many common symptoms like hot flushes, night sweats, vaginal dryness, and changes in mood or sleep patterns. It also has longer-term effects on bone density and cardiovascular health.
There was some discussion about hormone replacement therapy, HRT, wasn't there? Especially regarding heart disease risk.
Yes, that's an important point often discussed in HL biology. Early observational studies seem to show that women taking HRT, estrogens, sometimes with progesterone, had a lower risk of coronary heart disease, CHD. This led to widespread use.
But later studies showed something different.
Correct. Later, more rigorous randomized controlled trials, RCTs, like the Women's Health Initiative, actually indicated that HRT might slightly increase the risk of CHD, as well as stroke and breast cancer in some women, although it was effective for menopause symptoms.
So why the difference between the early observations and the controlled trials?
It turned out that socioeconomic status was a major confounding factor in the earlier studies. Women who chose to take HRT early on tended to be healthier, wealthier, and had better access to healthcare overall factors that independently reduce CHD risk. The initial correlation wasn't necessarily causation. It's a classic example of why controlled experiments are so important in science and medicine.
Correlation versus causation. A crucial distinction. Okay, just very briefly, let's loop back to sexual reproduction and variation. Meiosis is central.
Absolutely central. Meiosis isn't just about having chromosome numbers. It's a major engine of genetic variation in sexually reproducing organisms.
And how does it generate this variation? What are the key mechanisms?
There are three main sources linked to meiosis and fertilization. First, crossing over. During prophysi of meiosis, homologous chromosomes pair up and exchange segments of genetic material. This creates new combinations of alleles on each chromosome.
Swapping bits of chromosomes. What's the second?
Independent assortment. When homologous chromosome pairs line up at the cell's equator during metaphase I, how they orient which one faces which pole is random for each pair. This means the combination of maternal and paternal chromosomes that end up in each daughter cell, and thus each gamete, is shuffled randomly. With 23 pairs of chromosomes in humans, the number of possible combinations is huge, over 8 million.
Random shuffling of whole chromosomes. And the third source?
Random fertilization. Any one of the millions of genetically unique sperm produced has an essentially random chance of fertilizing any one of the genetically unique eggs produced, or the single egg released. This multiplies the potential for variation astronomically.
Crossing over, independent assortment, random fertilization, all working together to make unique offspring.
Precisely. That's why siblings, except identical twins, are genetically different from each other and from their parents. This variation is the raw material for evolution.
And just one last related topic, often covered. In vitro fertilization, or IVF. How does that work in brief?
IVF is an assisted reproductive technology. Essentially, it involves controlling the woman's hormone cycle, often using drugs to suppress natural hormones, and then administering high doses of hormones, like FSH, to stimulate the ovaries to produce multiple mature eggs at once. That's superovulation.
Getting multiple eggs instead of just one. Then what?
The mature eggs are retrieved from the ovaries surgically. Sperm is collected from the male partner or a donor. Fertilization is then performed in a laboratory dish. In vitro means in glass. Sometimes sperm are simply mixed with the eggs. Other times, especially if there are sperm issues, a single sperm might be injected directly into an egg using a technique called ICSI, intracytoplasmic sperm injection.
Fertilization in the lab. And then?
The fertilized eggs, zygotes, are cultured in the lab for a few days as they develop into embryos, typically to the blastocyst stage. Then, one or more selected embryos are transferred into the woman's uterus through the cervix, with the hope that at least one will implant in the endometrium and establish a successful pregnancy.
Okay, a complex process bypassing some of the natural steps. We've covered a massive amount of ground today. What are the absolute key takeaways for Mr. Martin's class to remember from this reproductive biology deep dive?
I'd say. Grasp the fundamental trade-off asexual reproduction offers speed and efficiency in stable conditions but lacks variation, while sexual reproduction is costly but generates the crucial genetic diversity needed for adaptation. Understand the intricate hormonal control, especially in the human menstrual cycle, coordinating ovarian and uterine events. Appreciate the specifics of fertilization preventing polyspermy in animals, the unique double fertilization in flowering plants. And finally, always link sexual reproduction back to meiosis and the generation of variation, which is fundamental to evolution.
It really is amazing how these processes ensure not just the continuation of life, but also its ability to change and diversify over time.
Absolutely. It underscores how reproduction is central to both the stability and the dynamism of the biological world.
Well, Mr. Martin's IBU biology class, we sincerely hope this deep dive has clarified these essential concepts for you. If you found it useful and you'd like more explorations like this, please do hit that like button, subscribe to our channel, and definitely please leave comments below any questions you still have or other biology topics you'd like us to tackle next time. We really want to hear from you.
And maybe just a final thought to chew on. Considering all the different strategies we discussed, budding, fission, flowers, internal fertilization, external fertilization, how does this sheer variety of reproductive methods itself contribute to the incredible biodiversity we see all around us on Earth? Something to ponder.